12 synaptic transmission begging with the immediate of two nerve impulses. This diagram shows a sequence of orbits involved in synaptic transmission at the neuromuscle's large direction. A typical chemical synapse. E orbits shown here are for acetylcholine or similar for other neurotransmitters. Myelin. After synaphase transmission, ANET, RID1. After synaphase transmission, the increase in CA inside the axon terminal causes the vesicles containing acetylcholine to fuse with the presynaptic membrane and empty their contents into the synaptic cleft. The acetylcholine molecules diffuse within the cleft and bind to acetylcholine receptors on the motor end plate. The postsynaptic membrane integrates synaptic input. The postsynaptic membrane of the neuromuscular junction differs from the presynaptic membrane in an important way. Motor end plates have very few voltage-gated sodium channels, therefore, they do not fire action potentials. This is true not only of motor end plates on muscle cells, but also of most dendrites in most regions of neuronal cell bodies. The binding of acetylcholine to receptors at the motor end plate and the opening of chemically gated ion channels produce a change in the membrane potential of the postsynaptic membrane. This local change in membrane potential spreads to neighboring regions of the plasma membrane of the postsynaptic cell. The entire plasma membrane of a muscle cell, except for the motor end plates, contains voltage-gated sodium channels. If the axon terminal of a motor neuron releases sufficient amounts of acetylcholine to depolarize a motor end plate enough, that spreading depolarization will reach an area of Institute for Advanced Studies, Jodhpur 45. Unit 7 System Physiology Animal. Plasma membrane that contains voltage-gated sodium channels. When that area of membrane is depolarized to threshold, an action potential is fired. This action potential is then conducted throughout the muscle cell's system of membranes, causing the cell to contract. How much neurotransmitter is enough? Neither a single acetylcholine molecule nor the contents of an entire vesicle, about 10,000 acetylcholine molecules, are enough to bring the plasma membrane of a muscle cell to threshold. However, a single action potential in an axon terminal releases the contents of about 100 vesicles, which is enough to fire an action potential in the muscle cell and cause it to contract. Synapses between neurons can be excitatory or inhibitory. In vertebrates, the synapses between motor neurons and muscle cells are always excitatory. That is, motor end plates always respond to acetylcholine by depolarizing the postsynaptic membrane. Synapses between neurons, however, are not always excitatory. Recall that a neuron may have many dendrites. Axon terminals from many other neurons may form synapses with those dendrites and with the cell body. The axon terminals of different presynaptic neurons may store and release different neurotransmitters, and the plasma membrane of the dendrites and cell body of a postsynaptic neuron may have receptors for a variety of neurotransmitters. Thus, at any one time, a postsynaptic neuron may receive area 4. Decision making is the axon hillock, the region of the cell body at the base of the axon. See figure 2. The plasma membrane of the axon hillock is not insulated by glial cells and has many voltage-gated channels. Excitatory and inhibitory postsynaptic potentials from synapses anywhere on the dendrites of the cell body spread to the axon hillock by local current flow. If the resulting combined potential depolarizes the axon hillock to threshold, the axon fires an action potential. Because postsynaptic potentials decrease in strength as they spread from the site of the synapse, all postsynaptic potentials do not have equal influences on the axon hillock. A synapse at the tip of a dendrite has less influence than a synapse on the cell body near the axon hillock. Excitatory and inhibitory postsynaptic potentials can be summed over space or over time. Spatial summation adds up the simultaneous influences of synapses at different sites on the postsynaptic cell. Figure 15a. Temporal summation adds up postsynaptic potentials generated at the same site in a rapid sequence. Figure 15b. Excitatory. Dendrites equals 3. Synapse vessel. Dash. Action K. Potential variety of different chemical messages. If the spatial summation occurs, temporal summation means postsynaptic neurons response to a neurotransmitter is equals W. T's. 5 feet. 1 foot. 2 feet. Individual neurons can decide whether or not to fire an action potential by summoning excitatory and inhibitory postsynaptic potentials. This summation ability is a major mechanism by which the nervous system integrates information. Each neuron may receive 1,000 or more synaptic inputs, but it has only one output, an action potential in a single axon. All the information contained in all the inputs a neuron receives is reduced to the rate at which that neuron generates nerve impulses in its axon. All the information contained in all the inputs a neuron receives is reduced to the rate at which that neuron generates nerve impulses in its axon. For most neurons, the critical 15. The postsynaptic neuron sums information. Individual neurons sum excitatory and inhibitory postsynaptic potentials over space and time p. When the sum of the potentials depolarizes the axon hillock to threshold, the neuron generates an action potential. All the neuron-to-neuron synapses that we have discussed up to this point are between the axon terminals of a presynaptic cell and the cell body or dendrites of a postsynaptic cell. Synapses can also form between the axon terminals of one neuron and the axon terminals of another neuron. Such a synapse can modulate how much neurotransmitter the second neuron releases in response to action potentials traveling down its axon. We refer to this mechanism of regulating synaptic strength as presynaptic excitation or presynaptic inhibition. There are two types of neurotransmitter receptors. Most neurotransmitter receptors induce changes in postsynaptic cells by opening or closing ion channels. How they do so is the basis for grouping receptors into two general categories. The ionotropic receptors are themselves ion channels. Neurotransmitter binding by an ionotropic receptor causes a direct change in 46 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Ion movement across the plasma membrane of the postsynaptic cell. These proteins are also called ligand-gated channels. The metabotropic receptors are not ion channels, but they induce changes in the postsynaptic cell that can secondarily lead to changes in ion channels. Postsynaptic cell responses mediated by metabotropic receptors are generally slower and longer lasting than those induced by ionotropic receptors. The acetylcholine receptor of the motor end plate is an example of an ionotropic receptor. It consists of 5 subunits, each of which extends through the plasma membrane. See figure 14. When assembled, the subunits create a central pore that allows ions to pass through. There are several different kinds of subunits, and only one kind has the ability to bind acetylcholine. Each functional receptor has 2 of the acetylcholine-binding subunits and 3 other subunits. Metabotropic receptors are also transmembrane proteins, but instead of acting as ion channels, they initiate an intracellular signaling process that can result in the opening or closing of an ion channel. These receptors have 7 transmembrane domains, and they are linked to G proteins. Figure 16. See also figure 17. When a neurotransmitter binds to the extracellular domain of a metabotropic receptor, the intracellular domain activates a G protein. In its inactive state, the G protein has 3 subunits, one of which, the O subunit, is bound to a molecule of GDP. When the receptor binds its neurotransmitter, the GDP is replaced with a GTP molecule, and the O subunit separates from the other 2 subunits called B and Y. The A subunit moves laterally in the membrane until it binds to and opens an ion channel or binds to an effector protein that activates a second messenger cascade, which in turn opens an ion channel. Cube side of cell. L neurotransmitter binds one O the receptor. S O G protein IG protein O A subunit. AG protein subunit activates an ion channel directly or indirectly through a second messenger. The receptor activates AG protein. Inside of cell. 16 metabotropic receptors act through G proteins. Metabotropic receptors activate G proteins, which can influence ion channels directly or through second messengers. Electrical synapses are fast but do not integrate information well. Electrical synapses are different from chemical synapses because they couple neurons electrically. Electrical synapses are gap junctions. At these synapses, the presynaptic and postsynaptic cell membranes are separated by a space of only 2 to 3 nanometers, and specific membrane proteins called connexons link the 2 neurons by forming molecular tunnels between the 2 cells. Ions and small molecules can pass directly from cell to cell through the connexons. Transmission at electrical synapses is very fast and can proceed in either direction, whereas transmission at chemical synapses is slower and unidirectional. Electrical synapses are less common in the nervous systems of vertebrates than are chemical synapses for several reasons. First, electrical continuity between neurons does not allow temporal summation of synaptic inputs. Second, an effective electrical synapse requires a large area of contact between the presynaptic and postsynaptic cells. This condition rules out the possibility of thousands of synaptic inputs to a single neuron, which is the norm in complex nervous systems. Third, electrical synapses cannot be inhibitory. Finally, chemical synapses appear to have considerable plasticity, modifiability, as we will see below, but electrical synapses do not. Thus, electrical synapses are useful for rapid communication, such as that between muscle cells. But they are less useful for processes of integration and learning. The action of a neurotransmitter depends on the receptor to which it binds. More than 25 neurotransmitters are now recognized, and more will surely be discovered. Table 1 describes some of the best known neurotransmitters. Acetylcholine, as we have seen, is an important neurotransmitter because it is a means whereby the nervous system commands muscles to contract. Acetylcholine also plays roles in certain synapses between neurons in the central nervous system, but it accounts for only a small percentage of the total neurotransmitter content of the CNS. The workhorse neurotransmitters of the CNS are simple amino acids, glutamate excitatory, and glycine and GABA inhibitory. Another important group of neurotransmitters in the CNS is the monoamines, which are derivatives of amino acids. They include dopamine and norepinephrine, derivatives of tyrosine, and serotonin, a derivative of tryptophan. Peptides also function as neurotransmitters. An exciting recent discovery revealed that two gases, carbon monoxide and nitric oxide, are used by neurons as intercellular messengers even though they do not have the characteristics of classic neurotransmitters, that is, they do not have receptors. See figure 13. The complexity of neurotransmission is increased by the fact that each neurotransmitter has multiple receptor types. Acetylcholine, for example, has two receptor types, nicotinic receptors, which are ionotropic, and muscarinic receptors, which are metallotropic. Both types of acetylcholine receptors are found in the CNS, where nicotinic receptors tend to be excitatory and muscarinic receptors tend to be inhibitory. Acetylcholine actions can differ outside of the CNS as well. Acetylcholine acting through nicotinic receptors causes the smooth muscle of the gut to depolarize and therefore increases its motility, but acetylcholine acting through muscarinic receptors causes cardiac muscle to hyperpolarize and therefore decreases the contractility of the heart. Institute for Advanced Studies, Jodhpur 47. Unit 7 System Physiology Animal. Some well-known neurotransmitters. NEURGTRANSMITT. Some people have reactions to the food additive monosodium glutamate because it can affect the nervous system. Drugs called benzodiazepines, used to reduce anxiety and produce sedation, mimic the actions of GABA. Receptors are activated by narcotic drugs, opium, morphine, heroin, codeine. MOA classic neurotransmitter. It diffuses across membranes rather than being released synaptically. A means whereby a postsynaptic cell can influence a presynaptic cell. We could give many more examples of neurotransmitters that have different effects in different tissues, but the important thing to remember is that the action of a neurotransmitter depends on the receptor to which it binds. Glutamate receptors may be involved in learning and memory. Glutamate is a neurotransmitter that can bind to a variety of receptors, including both metallotropic and ionotropic receptors. The glutamate receptors are divided into several classes because they can be differentially activated by other chemicals that mimic the action of glutamate. One class of ionotropic glutamate receptors is the NMDA receptors, which can be activated by the chemical N-methyl D-aspartate. Another class of ionotropic glutamate receptors is activated by a different chemical, abbreviated as AMPA. Glutamate is an excitatory neurotransmitter, so activation of glutamate receptors always results in non-entry into the neuron and depolarization. But the timing of the response to activation by these different types of receptors differs significantly. The AMPA receptors allow rapid influx of Na into the postsynaptic cell, while the NMDA receptors allow slower and longer-lasting influx of Na. The NMDA receptors also require that the cell be somewhat depolarized through the action of other receptors before their pores will open and permit Na influx. When they do open, these receptors also allow Ca to enter the cell. Ca ions act as second messengers in the cell and can trigger a variety of long-term cellular changes. Most of the synaptic events we have studied so far happen very quickly. It is therefore a special challenge to 48 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Understand how the messages carried by action potentials can result in long-term events such as learning and memory. Our understanding of these processes has been greatly affected by a phenomenon called long-term potentiation, or LTP, that was discovered by neurobiologists working with slices of brain kept alive in dishes of culture medium. Using these brain slice preparations, it is possible to stimulate and record from specific brain regions, or even specific neurons. In the studies leading to the discovery of LTP, experimenters repeatedly stimulated synaptic inputs to a particular neuron and observed the usual action potential response. When the neuron was stimulated many times in rapid succession, however, they found that the properties of the experiment neuron changed. The magnitude of the postsynaptic response was enhanced, or potentiated, and this change lasted for days or weeks. How does this modification of a synapse occur? The answer in some areas of the brain now seems quite clear. With low levels of stimulation, the glutamate released by presynaptic cells activates only the AMPA receptors, and the postsynaptic membrane simply responds with action potentials. With higher levels of stimulation, however, the NMDA receptors are activated, allowing both Na+ and Ca2+ ions to enter the postsynaptic neuron. The Ca ions induce long-term changes in the postsynaptic membrane that make it more sensitive to synaptic input. Figure 18. Question: Can stimulation change the properties of a synapse? Method: Chelly's automatically stimulated through results to input pathways and responses recorded. Response 2. Stimulating electrode 1. Stimulating sternulating. Electrode 1 electrode. Conclusion: high-frequency stimulation of synapses can cause a long-lasting change in the sensitivity of these synapses. Long-term potentiation. 18. Repeated stimulation can cause long-term potentiation when a cell receives regular synaptic input. The resulting postsynaptic potential remains constant. If, however, that same synaptic pathway is stimulated briefly at a high-frequency TH, the subsequent sensitivity of the postsynaptic cell to the original level of synaptic input is potentiated for a long time. Exploiting the LTP system, Dr. Joe Chen and his students and collaborators at Princeton University genetically engineered mice so that their NMDA receptors had a slightly altered structure and were activated for a longer time whenever they bound a molecule of glutamate. These mice learned tasks better, ran mazes faster, and remembered the mazes longer than normal mice. These exciting experiments show that we are on the right track to understanding how the brain achieves learning and memory. To turn off responses, synapses must be cleared of neurotransmitter. Turning off the action of neurotransmitters is as important as turning it on. If released neurotransmitter molecules simply remained in the synaptic cleft, the postsynaptic membrane would become saturated with hyperpolarized or depolarized and would be unresponsive to short-term changes in the presynaptic cell. The more discrete each separate neuronal signal is, the more information can be processed in a given time. Thus, neurotransmitter must be cleared from the synaptic cleft shortly after it is released by the axon terminal. Neurotransmitter action may be terminated in several ways. First, enzymes may destroy the neurotransmitter. Acetylcholine, for example, is rapidly destroyed by the enzyme acetylcholinesterase, which is present in the synaptic cleft in close association with the acetylcholine receptors on the postsynaptic membrane. See figures 13 and 14. Some of the most deadly nerve gases developed for chemical warfare work by inhibiting acetylcholinesterase. As a result, acetylcholine lingers in the synaptic clefts, causing the victim to die of spastic neurotransmitter, and receptors would be constantly contracted, muscle paralysis. Some agricultural activated. As a result, the postsynaptic cell would remain insecticides, such as malathion, also inhibit. Institute for Advanced Studies, Jodhpur 49. Unit 7 System Physiology Animal. Acetylcholinesterase and can poison farm workers if used without safety precautions. Second, neurotransmitter may simply diffuse away from the cleft. Third, neurotransmitter may be taken up via active transport by nearby cell membranes. Prozac, a drug commonly prescribed to treat depression, slows the reuptake of the neurotransmitter serotonin, thus enhancing its activity at the synapse. Because neurons can interact in the complex ways we have just discussed, networks of neurons can process and integrate information. Multiple neuronal networks constitute the nervous systems of animals. In the next sections, we will see many examples of how neurons work together in networks to accomplish specific tasks. These networks use all of the mechanisms we have discussed in this chapter: excitatory and inhibitory synapses, presynaptic excitation and inhibition, and mechanisms of long-term potentiation. Through these operations, our brains solve puzzles, create inventions, remember experiences, fall in love, and learn about biology. The challenge for the future one is to understand how these networks work. The nervous system: structure, function, and information flow. The human nervous system consists of three major components. The brain and spinal cord together constitute the central nervous system, CNS. Information is transmitted to and from the CNS by means of an enormous network of nerves that make up the peripheral nervous system, PNS. The PNS reaches every tissue of the body. It connects to the CNS via spinal nerves and cranial nerves. A nerve is a bundle of axons that carries information about many things simultaneously. It is important to distinguish between the axon of a single neuron and a nerve. Some axons in a nerve may be carrying information to the CNS, while other axons in the same nerve are carrying information from the CNS to the organs of the body. Neural efferents carry signals away from the CNS. Neural afferents bring signals to the CNS. Neural afferents efferents. O skeletal. Skeletal muscles conscious voluntary. Central nervous. L system. Unconscious. Aus CNS autonomic. Body peripheral glands, smoother, gans nervous muscle, heart. System muscle. PNS. Neural hormones. One organization of the nervous system, the peripheral nervous system, indicated by pink and blue, carries information both to and from the central nervous system. The CNS also receives hormonal inputs and produces hormonal outputs. A conceptual diagram of the nervous system traces information flow. The major avenues of information flow through the nervous system are illustrated in Figure 1. The afferent portion of the peripheral nervous system carries information to the CNS. We are consciously aware of much of the information that moves through these afferent pathways, for example, vision, hearing, temperature, pain, the position of limbs. We are not consciously aware of other afferent information that is important for physiological regulation, for example, blood pressure, deep body temperature, blood oxygen supply. The afferent portion of the peripheral nervous system carries information from the CNS to the muscles and glands of the body. Afferent pathways can be divided into a voluntary division, which executes our conscious movements, and an involuntary, or autonomic, division, which controls physiological functions. In addition to neuronal information, the CNS receives chemical information in the form of hormones circulating in the blood. Neural hormones released by neurons into the extracellular fluids of the brain can send chemical information to other neurons in the brain or can leave the brain and enter the circulation. Earlier we have learned important role of neural hormones such as GnRH in the control of the anterior pituitary, and we saw that other neural hormones such as oxytocin are released from the posterior pituitary into the circulation. The vertebrate CNS develops from the embryonic neural tube. Early in the development of all vertebrate embryos, a hollow tube of neural tissue forms. This neural tube runs the length of the embryo on its dorsal side. At the anterior end of the embryo, the neural tube forms three swellings that become the basic divisions of the brain: the hindbrain, the midbrain, and the forebrain. The rest of the neural tube becomes the spinal cord, Figure 2. The cranial and spinal nerves, which make up the peripheral nervous system, sprout from the neural tube and grow throughout the embryo. Each of the three regions of the embryonic brain develops into several structures in the adult brain. From the hindbrain come the medulla, the pons, and the cerebellum. The medulla is continuous with the spinal cord. The pons is in front of the medulla, and the cerebellum is the dorsal outgrowth of the pons. The medulla and pons contain distinct groups of neurons that are involved in the control of physiological functions such as breathing and circulation or basic motor patterns such as swallowing and vomiting. All information traveling between the spinal cord and higher brain areas must pass through the pons and the medulla. The cerebellum runs like the conductor of an orchestra. It receives copies of the commands going to the muscles from higher brain areas, and it receives information coming up the spinal cord from the joints and muscles. Thus it can compare the motor score with the actual behavior of the muscles and refine the motor commands. 50 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. From the embryonic midbrain come structures that process aspects of visual and auditory information. In addition, all information traveling between higher brain areas and the spinal cord must pass through the midbrain. The structures that develop from the hindbrain and the midbrain are collectively known as the brainstem. Shows three swellings that will form the adult brain. The stretched out neural tube, viewed from above. Lateral views, dorsal views. Neural tube. Forebrain. Midbrain. Hindbrain, I smiley face. Spinal cord, 1. 25 days. AF the forebrain develops into TOIO, AE, major divisions TH, E telencephalon. 35 days in diencephalon. Forebrain, midbrain, telencephalon. The hindbrain develops into three major divisions: the cerebellum, pons, and medulla. 2. Development of the human nervous system: three swellings at the anterior end of the hollow neural tube in the early vertebrate embryo develop into the parts of the adult brain. The final view is an adult human brain cut in half through the midline. The embryonic forebrain develops a central region called the diencephalon and a surrounding structure called the telencephalon. The diencephalon is the core of the forebrain and consists of an upper structure called the thalamus and a lower structure called the hypothalamus. The thalamus is the final relay station for sensory information going to the telencephalon, and the hypothalamus is responsible for the regulation of many physiological functions and biological drives. Adult brain. The telencephalon consists of two cerebral hemispheres, left and right, and is also referred to as the cerebrum. In humans, the telencephalon is by far the largest part of the brain and plays major roles in sensory perception, learning, memory, and conscious behavior. As we go up the vertebrate phylogenetic scale from fishes to mammals, the telencephalon increases in size, complexity, and importance. The forebrain dominates the nervous systems of mammals, and damage to this region results in severe impairment of sensory, motor, or cognitive functions, and even coma. In contrast, a shark, with its telencephalon removed, can swim almost normally. Functional subsystems of the nervous system. We have just surveyed the development of the nervous system in terms of anatomically distinct structures. At any one time, these various structures are engaged in many simultaneous tasks, a property known as parallel processing of information. Specific tasks are carried out by subsystems that may involve several different anatomical regions or structures of the nervous system. We will now examine several of these functional subsystems. The spinal cord receives and processes information from the body. The spinal cord conducts information in both directions between the brain and the organs of the body. It also integrates a great deal of the information coming from the peripheral nervous system, and it responds to that information by issuing motor commands. The spinal cord serves two important functions in an animal. It is the connecting link between the brain and most of the body, and it is involved in spinal reflex actions. A reflex is a predictable, involuntary response to a stimulus. Thus, both voluntary and involuntary limb movements, as well as certain organ functions, depend on this link. The spinal cord is the part of the central nervous system that extends from the brain to near or into the tail. A cross-section shows a neural canal that contains cerebrospinal fluid. The gray matter consists of cell bodies and dendrites, and is concerned mainly with reflex connections at various levels of the spinal cord. Extending from the spinal cord are the ventral and dorsal roots of the spinal nerves. These roots contain the main motor and sensory fibers, axons and/or dendrites respectively, that contribute to the major spinal nerves. The white matter of the spinal cord gets its name from the whitish myelin that covers the axons. 3 layers of protective membranes called meninges place. Of Mennings, membranes surround the spinal cord. They are continuous with similar layers that cover the brain. The outer layer, the dura mater, is a tough, fibrous membrane. The middle layer, the arachnoid, is delicate and connects to the innermost layer, the pia mater. The pia mater contains small blood vessels that nourish the spinal cord. Spinal nerves. Generally, the number of spinal nerves is directly related to the number of segments in the trunk and tail of a vertebrate. For example, a frog has. Institute for Advanced Studies, Jodhpur 51. Unit 7 System Physiology Animal. Evolved strong hind legs for swimming or jumping, a reduced trunk, and no tail in the adult. It has only 10 pairs of spinal nerves. By contrast, a snake, which moves by lateral undulations of its long trunk and tail, has several hundred pairs of spinal nerves. Reticular system. The reticular system is a highly complex network of axons and dendrites. Within the reticular system are many discrete groups of neurons. Such an anatomically distinct group of neurons in the CNS is called a nucleus, not to be confused with the nucleus of a single cell. The reticular system is distributed through the core of the medulla, pons, and midbrain. Afferent information passes through the reticular system, where many connections are made to neurons involved in controlling many functions of the body. Information from joints and muscles, for example, is directed to nuclei in the pons and cerebellum that are involved in balance and coordination, whereas information from pain receptors is directed to nuclei that control sensitivity to pain. This information continues upward to the forebrain, where it results in conscious sensations that can be localized to the specific sites in the body where the information originated. The information routed through the reticular system also influences the level of arousal of the nervous system. Nuclei in the reticular system are involved in the control of sleep and waking. High levels of activity in the reticular system influence these nuclei to maintain the brain in a waking condition. Low levels of activity enable sleep. Because of the alerting function of the reticular core of the brain stem, it has been called the reticular activating system. If the brain of a person is damaged at midbrain or higher levels and the alerting action of the reticular system cannot reach the forebrain, the person loses the ability to be in a conscious, waking state and becomes comatose. Damage to the brain stem or the spinal cord below the reticular system does not interfere with the ascending alerting actions of the reticular system and leaves the person with normal patterns of sleep and waking. However, such damage can cause loss of sensation and loss of motor function. The limbic system supports basic functions of the forebrain. The telencephalon of fishes, amphibians, and reptiles consists of only a few structures surrounding the diencephalon. In birds and mammals, these primitive forebrain structures are completely covered by the evolutionarily more recent elaborations of the telencephalon called the neocortex, but these primitive forebrain structures still have important functions. These structures are collectively referred to as the limbic system, figure 3. The limbic system is responsible for basic physiological drives, instincts, and emotions. Within the limbic system are areas that, when stimulated with small electric currents, can cause intense sensations of pleasure, pain, or rage. If a rat is given the opportunity to stimulate its own pleasure centers by pressing a switch, it will ignore food, water, and even sex, pushing the switch until it is exhausted. Pleasure and pain centers in the limbic system are believed to play roles in learning and in physiological drives. Pleasure and pain centers in the limbic system are believed to play roles in learning and in physiological drives. Structures deep within the cerebral hemispheres and surrounding the hypothalamus control aspects of motivation, drives, emotions, and memory. Cerebral. 3. The limbic SV stem. The evolutionarily primitive parts of the telencephalon are referred to as the limbic system. A component of the limbic system, the amygdala, is involved in fear and fear memory. If a certain portion of the amygdala is damaged or chemically blocked, an animal cannot learn to be afraid of a stimulus or a situation that would normally induce a strong fear reaction. Moreover, blocking protein synthesis in this part of the limbic system blocks the formation of fear memory. Another part of the limbic system, the hippocampus, is necessary in humans for the transfer of short-term memory to long-term memory. If you are told a new telephone number, you may be able to hold it in short-term memory for a few minutes, but within half an hour it is forgotten unless you make a real effort to remember it. The phenomenon of remembering something for more than a few minutes requires its transfer to long-term memory. Regions of the cerebrum interact to produce consciousness and control behavior. The cerebral hemispheres are the dominant structures in the mammalian brain. In humans, they are so large that they cover all other parts of the brain except the cerebellum, figure 4a. A sheet of gray matter called the cerebral cortex covers each cerebral hemisphere. It is about 4 millimeters thick and covers a total surface area over both hemispheres of 1 square meter. The cerebral cortex is convoluted, or folded, into ridges called gyrite, singular, gyrus, and valleys called sulci, singular, sulcus. These convolutions allow it to fit into the skull. Under the cerebral cortex is white matter, made up of the axons that connect the cell bodies in the cortex with one another and with other areas of the brain. A curious feature of our nervous systems is that the left side of the body is served in both sensory and motor aspects, mostly by the right side of the brain, and the right side of the body is served mostly by the left side of the brain. Thus, sensory input from the right hand goes to the left cerebral hemisphere, and sensory input from the 52 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Left hand goes to the right cerebral hemisphere. The two hemispheres, however, are not exactly symmetrical. Language abilities, for example, reside predominantly in the left hemisphere, as we will see below. Different regions of the cerebral cortex have specific functions, figure 4b. Some of those functions are easily defined, such as receiving and processing sensory information, but most of the cortex is involved in higher order information processing that is less easy to define. These latter areas are given the general name of association cortex. To understand the cerebral cortex, it helps to have an anatomical roadmap. As viewed from the left side, the left cerebral hemisphere looks like a boxing glove for the right hand with the fingers pointing forward, the thumb pointing out, and the wrist at the rear, see figure 4a. The thumb area is the temporal lobe, the fingers the frontal lobe, the back of the hand the parietal lobe, and the wrist the occipital lobe. A mirror image of this arrangement characterizes the right cerebral hemisphere. Let's look at each lobe of the cerebrum separately. At central sulcus, olfactory bulb, temporal lobe, cerebellum, spinal cord. Viewed here from the left side, covers all the highly convoluted cerebral cortex of the other structures of the forebrain. For the human cerebrum, each cerebral hemisphere is divided into 4 lobes. These different functions are localized in particular areas of the cerebral lobes. Central primary motor cortex, primary somato, and motor control, sensory cortex, touch and pressure. The temporal lobe. The upper region of the temporal lobe receives and processes auditory information. The association areas of the temporal lobe are involved in the recognition, identification, and naming of objects. Damage to the temporal lobe results in disorders called agnosis, in which the individual is aware of a stimulus but cannot identify it. Damage to one area of the temporal lobe results in the inability to recognize faces. Even old acquaintances cannot be identified by facial features, although they may be identified by other attributes such as voice, body features, and characteristic style of walking. Using monkeys, it has been possible to record the activity of neurons in this region that respond selectively to faces in general, figure 5. These neurons do not respond to other stimuli in the visual field, and the responsiveness decreases if some of the features of the face are missing or appear in inappropriate locations. Damage to other association areas of the temporal lobe causes deficits in understanding spoken language, even though speaking, reading, and writing abilities may be intact. This neuron responds maximally to a complete face view from the front. FIRNG. R8A. 5 neurons in one region of the temporal lobe respond to faces. The traces represent the firing rate of a neuron in the temporal lobe of a monkey in response to the pictures shown below them. The frontal lobe. The frontal and parietal lobes are separated by a deep valley called the central sulcus. Asterisk of the frontal lobe cortex just in front of the central sulcus is called the primary motor cortex, see figure 4b. The neurons in this region control muscles in specific parts of the body. The parts of the body can be mapped onto the primary motor cortex, from the head region on the lower side to the lower part of the body at the top. Areas with fine motor control, such as the face and hands, have the greatest representation, figure 6. Somatosensory. The left side of the cerebral Cortex communicates with the right side of the body, and vice versa. Motor cortex, somatosensory cortex. 6. The body is represented in the primary motor cortex and the primary somatosensory cortex cross-sections through the primary motor and primary somatosensory cortexes can be represented as maps of the human body. Body parts are shown in proportion to the brain area devoted to them. Institute for Advanced Studies, Jodhpur 53. Unit 7 System Physiology Animal. If a neuron in the primary motor cortex is electrically stimulated, the response is a twitch of a muscle, but not a coordinated, complex behavior. The association functions of the frontal lobe are diverse. They are best described as having to do with planning, and they contribute very significantly to personality. People with frontal lobe damage have drastic alterations of personality because they cannot create an accurate view of themselves in the context of the world around them and cannot plan for future events. A dramatic case of frontal lobe damage is the story of Phineas Gage, who was an industrious, responsible, considerate young railroad construction foreman in 1848. Then a blasting accident shot a meter-long, 3-centimeter wide iron tamping rod through his brain. The tamping iron entered Gage's head below his left eye, passed through his frontal lobe, and exited the top of his head. Figure 7. 7 mind-altering experience in a 19th century railroad construction accident. An explosion blew a tamping iron through the brain of Phineas Gage. Unbelievably, Gage survived, but his personality was radically changed. This drawing of Gage's skull was made at the time of his death. Remarkably, Gage survived this terrible accident, but he was a completely different person. He was quarrelsome, bad-tempered, lazy, and irresponsible. He was impatient and obstinate, and he used profane language, which he had never done before. He spent the rest of his days as a drifter, earning money by telling his story, exhibiting his scars and the tamping iron. If you are in Cambridge, Massachusetts, you can pay him a visit. His skull, death mask, and the tamping iron are on display in the Museum of the Medical College of Harvard University. The parietal lobe. The strip of parietal lobe cortex just behind the central sulcus is the primary somatosensory cortex. See figure 4b. This area receives touch and pressure information through the thalamus. The whole body surface can be mapped onto the primary somatosensory cortex. See figure 6. Areas of the body that have a high density of tactile mechanoreceptors and are capable of making fine discriminations in touch, such as the lips and the fingers, have disproportionately large representation. If a very small area of the primary somatosensory cortex is stimulated electrically, the subject reports feeling specific sensations, such as touch, in a very localized part of the body. A major association function of the parietal lobe is attending to complex stimuli. Damage to the right parietal lobe causes a condition called contralateral neglect syndrome, in which the individual tends to ignore stimuli from the left side of the body or the left visual field. Such individuals have difficulty performing complex tasks, such as dressing the left side of the body. An afflicted man may not be able to shave the left side of his face. When asked to copy simple drawings, a person who exhibits this syndrome can do well with the right side of the drawing, but not the left. Figure 8. The parietal cortex is not symmetrical with respect to its role in attention. However, damage to the left parietal cortex does not cause the same degree of neglect of the right side of the body. We will see similar asymmetries in cortical function when we discuss language. Pound. Model and T_patients copy. A contralateral neglect syndrome. A person with damage to the right parietal association cortex will neglect the left side of a drawing when asked to copy a model. The occipital lobe. The occipital lobe receives and processes visual information. We'll learn more about the details of that process later in this chapter. The association areas of the occipital cortex are essential for making sense of the visual world and translating visual experience into language. Some deficits resulting from damage to these areas are specific. In one case, a woman with limited damage was unable to see motion. Her vision was intact, but she could see a waterfall only as a still image, and a car approaching only as a series of scenes of a stationary object at different distances. The cerebrum has increased in size and complexity. As mentioned earlier, the size of the telencephalon relative to the rest of the brain increases substantially as we move up the phylogenetic scale from fishes to amphibians, to reptiles, to birds and mammals. Even when we consider only mammals, the cerebral cortex increases in size and complexity when we compare animals such as rodents, whose behavioral repertoires are relatively simple, with animals such as primates that have much more complex behavior. The most dramatic increase in the size of the cerebral cortex took place during the last several million years of human evolution. The incredible intellectual capacities of Homo sapiens are associated with this enlargement of the cerebral cortex. Humans do not have the largest brains in the animal kingdom. Elephants, whales, and porpoises have larger brains in terms of mass. If we compare brain size to body size, however, humans and dolphins top the list. Humans have the largest ratio of brain size to body size, and they have the most highly developed cerebral cortex. Another feature of the cerebral cortex that reflects increasing behavioral and intellectual capabilities is the ratio of association cortex to primary somatosensory and motor cortexes. Humans have the largest relative amount of association cortex. 54 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Information processing by neuronal networks. The functions of the nervous system can be understood in terms of neuronal networks. In this section we will use two subsystems of the nervous system to demonstrate the functioning of such networks. The first example, the autonomic nervous system, consists of efferent pathways. The second, the visual system, consists of afferent and integrated pathways. Techniques that have allowed neurobiologists to trace neuronal connections, identify neurotransmitters at synapses, and record action potentials in single cells and groups of cells have advanced our understanding of how certain subsystems of the nervous system work. Parasympathetic division. Constricts pupils. Stimulates salivation, constricts airways. Cervical. Slows. Heartbeat stimulates. Digestion thoracic less than. Stimulates release of glucose. Stimulates halator. Stimulates activity of intestines. Stimulates urinary bladder bow contract. 7-5 feet. 1. The autonomic nervous system controls the physiological functions of organs and organ systems. The autonomic nervous system is divided into two parts, the sympathetic and parasympathetic divisions. These two divisions work in opposition to each other in their effects on most organs, one causing an increase in activity and the other causing a decrease. The two divisions of the autonomic nervous system are easily distinguished from each other by their anatomy, their neurotransmitters, and their actions. Figure 9. SV empathetic division. Dilates. Pupil. Inhibits. Salivation relaxes, always accelerates. Heartbeat. Inhibits digestion, inhibits release. O of glucose. FA, S nihilates, gallbladder. EY. ILE, FL inhibits activity. U stimulates fitter equals of intestines. Secretion of OS epinephrine, norepinephrine. Relaxes. O urinary. F bladder. O. 9. Organization of the autonomic nervous system. The autonomic nervous system is divided into the sympathetic and parasympathetic divisions, which work in opposition to each other in their effects on most organs, one causing an increase and the other a decrease in activity. Institute for Advanced Studies, Jodhpur 55. Unit 7 System Physiology Animal. The best known functions of the autonomic nervous system are those of the sympathetic division that produce the fight or flight response, increasing heart rate, blood pressure, and cardiac output and preparing the body for emergencies. In contrast, the parasympathetic division slows the heart and lowers blood pressure. It is tempting to think of the sympathetic division as the one that speeds things up and the parasympathetic division as the one that slows things down, but that is not always a correct distinction. The sympathetic division slows the digestive system, and the parasympathetic division accelerates it. Both divisions of the autonomic nervous system are efferent pathways. Each autonomic efferent pathway begins with a cholinergic neuron, one that uses acetylcholine as its neurotransmitter that has its cell body in the brainstem or spinal cord. These cells are called preganglionic neurons because the second neuron in the pathway with which they synapse resides in a ganglion, a collection of neuronal cell bodies that is outside of the CNS. The second neuron is called a postganglionic neuron because its axon extends out from the ganglion. The axon of the postganglionic neuron synapses with cells in the target organs. The postganglionic neurons of the sympathetic division are noradrenergic, use norepinephrine as their neurotransmitter, while the postganglionic neurons of the parasympathetic division are cholinergic. In organs that receive both sympathetic and parasympathetic input, the target cells respond in opposite ways to norepinephrine and to acetylcholine. A region of the heart called the pacemaker, which generates the heartbeat, is an example. Stimulating the sympathetic nerve to the heart or dripping norepinephrine onto the pacemaker region depolarizes the pacemaker cells, increases their firing rate, and causes the heart to beat faster. Stimulating the parasympathetic nerve to the heart or dripping acetylcholine onto the pacemaker region hyperpolarizes the pacemaker cells, decreases their firing rate, and causes the heart to beat more slowly. In contrast, in the digestive tract, norepinephrine hyperpolarizes muscle cells, which slows digestion, and acetylcholine depolarizes muscle cells, which accelerates digestion. The sympathetic and parasympathetic divisions of the autonomic nervous system can also be distinguished by anatomy. See Figure 9. The preganglionic neurons of the parasympathetic division come from the brainstem and the last segment of the spinal cord, the sacral region. The preganglionic neurons of the sympathetic division come from the upper regions of the spinal cord below the neck, the thoracic and lumbar regions. Most of the ganglia of the sympathetic division are lined up in two chains, one on either side of the spinal cord. The parasympathetic ganglia are close to, sometimes sitting on, the target organs. The autonomic nervous system is an important link between the CNS and many physiological functions of the body. Its control of diverse organs and tissues is crucial to homeostasis. In spite of its complexity, work by neurobiologists and physiologists over many decades has made it possible to understand its functions in terms of neuronal properties and circuits. For example, we will see how information from pressure receptors in the blood vessels is transmitted to the CNS, where it produces autonomic signals that control the rate of the heartbeat. Neurons and circuits in the occipital cortex integrate visual information. We have learned that the information conveyed to the brain in the optic nerve consists of action potentials that are stimulated by light falling on small circular areas of the retina called receptive fields. A receptive field contains many photoreceptor cells connected together in a circuit in such a way that the signals they produce are integrated and transmitted to the brain by a single retinal ganglion cell. The axon of each ganglion cell travels to the brain in the optic nerve. How does the brain construct visual I1 images from this information about circular patches of light falling on the retina? Information from the retina is transmitted through the optic nerve to a relay station in the thalamus, and then to the brain's visual processing area, in the occipital cortex at the back of the cerebral hemisphere. See Figure 4b. David Hubel and Torsten Diesel of Harvard University studied the activity of neurons in this visual cortex. They recorded the activities of single cells in the brains of living animals while they stimulated the animals' retinas with spots and bars of light. They found that cells in the visual cortex, like retinal ganglion cells, have receptive fields, specific areas of the retina that, when stimulated by light, influence the rate at which the cells fire action potentials. Cells in the visual cortex, however, have receptive fields that differ from the simple circular receptive fields of retinal ganglion cells. Cortical cells called simple cells are maximally stimulated by bars of light that have specific orientations. Simple cells probably receive input from several ganglion cells whose circular receptive fields are lined up in a row. Complex cells in the visual cortex are also maximally stimulated by a bar of light with a particular orientation, but the bar may fall anywhere on a large area of retina described as that cell's receptive field. The receptive field of a complex cell appears to be built from the receptive fields of several simple cells that share certain stimulus orientation, but have receptive fields in different places on the retina. Figure 10. Some complex cells respond most strongly when the bar of light moves in a particular direction. The concept that emerges from these experiments is that the brain assembles a mental image of the visual world by analyzing edges and patterns of light falling on the retina. This analysis is conducted in a massively parallel fashion. Each retina sends a million axons to the brain, but there are hundreds of millions of neurons in the visual cortex. Each bit of information from a retinal ganglion cell is received by hundreds of cortical cells, each responsive to a different combination of orientation, position, and even movement of contrasting lines in the pattern of light falling on the retina. Cortical cells receive input from both eyes. How do we see objects in three dimensions? The quick answer is that our two eyes, located at the front of the head, see overlapping, yet slightly different, visual fields. That once we have binocular vision. A person who is blind in one eye has great difficulty discriminating distances. Animals whose eyes are on the sides of the head have minimal overlap in their fields of vision and, as a result, 56 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Poor depth vision, but they can see predators creeping up from all sides. Experiment. Question. How do cells in the visual cortex respond to patterns of light falling on the retina? Method. The bear of light, a moving bar of light. As the cat views the screen, the moves across stimulates receptive electrode records activity and single the screen. Fields in the retina. Cells in the occipital cortex. Results. On-center ganglion. Cell receptive field. On-center ganglion. Cell response retinal ganglion cells that are AlloNet project to thalamic relay cells that relay information to cortex. Simple cells in the cortex. Respond to a static bear of light at a particular angle and location. Complex cells in the cortex. Respond to a moving bar epithelial. Columns. These columns alternate according to the source of their input. Left eye, right eye, left eye, right eye, and so on. Cells closest to the border between two columns receive input from both eyes and are therefore called binocular cells. Binocular cells interpret distance by measuring the disparity between where the same stimulus falls on the two retinas. Human brain viewed from underneath. Relay areas axons from thalamus, relay areas to visual cortex. The optic nerves cross at the optic chiasm. The visual cortex is organized in columns that receive input from the right eye yellow and the left eye blue. Interpretation. This model would explain the results of cell-to-cell connections. Columns receive input from both the right and left eyes. JF binocular cells at the borders of O X X X X X J1 The anatomy of binocular vision. Each eye transmits info match into both sides of the brain. However, the right side of the brain processes all information from the left visual field, and the left side of the brain processes all information from the right visual field. The visual cortex sorts visual field information according to whether it comes from the right eye or the left eye. Underscore. Relay cells, simple cells, complex cells. Retinal ganglion cells in thalamus, in cortex, in cortex. Conclusion. Cells in the retina, thalamus, and cortex are connected in such a way as to respond to specific patterns of light. What is disparity? Hold your finger out in front of you and look at it, closing one eye and then the other. Your finger appears to jump back and forth because its image falls on a different position on each retina. Repeat the exercise with an object at a distance. It doesn't appear to. 10 receptive fields of cells in the visual cortex. Cells in the visual cortex respond to specific patterns of light falling on the retina. Ganglion cells that transmit information about circular receptive fields converge on simple cells in the cortex in such a way that the simple cells have linear receptive fields. Simple cells transmit information to complex cells in such a way that the complex cells can respond to linear stimuli falling on different areas of the retina. The story of how the brain integrates information from two eyes begins with the paths of the optic nerves. If you look at the underside of the brain, the optic nerves from the two eyes appear to join together just under the hypothalamus and then separate again. The place where they join is called the optic chiasm. Figure 11. Axons from the half of each retina closest to your nose cross in the optic chiasm and go to the opposite side of your brain. The axons from the other half of each retina go to the same side of the brain. The result of this division of axons in the optic chiasm is that all visual information from your left visual field, everything left of straight ahead, goes to the right side of your brain, as shown in red in figure 11. All visual information from your right visual field goes to the left side of your brain, as indicated in green in the figure. Both eyes transmit information about a specific spot in your right visual field, for example, to the same place in the left visual cortex. Cells in the visual cortex are organized and jump back and forth as much because there is less disparity in the positions of the image on the two retinas. Certain binocular cells respond optimally to a stimulus falling on both retinas with a particular disparity. Which set of binocular cells is stimulated depends on how far away the stimulus is. When we look at something, we can detect its shape, color, depth, and movement. Where does all this information come together? Is there a single cell that fires only when a red sports car drives by? Probably not. A specific visual experience comes from simultaneous activity in a large collection of cells. In addition, most visual experiences are enhanced by information from the other senses and from memory as well. This realization helps explain why about 75% of the cerebral cortex is association cortex. Understanding higher brain functions in cellular terms. Very few functions of the nervous system have been worked out to the point of identifying the underlying. Institute for Advanced Studies, Job 57. Unit 7 System Physiology Animal. Neuronal networks. The processes responsible for the higher brain functions discussed in the remaining pages of this chapter are undeniably complex. Nevertheless, neurobiologists, using a wide range of techniques, are making considerable progress in understanding some of the cellular and molecular mechanisms involved in those processes. The following discussion presents several complex aspects of brain and behavior that present challenges to neurobiologists: sleep and dreaming, learning and memory, language use, and consciousness. Sleep and dreaming produce electrical patterns in the cerebrum. A dominant feature of human behavior is the daily cycle of sleep and waking. All birds and mammals, and probably all other vertebrates, sleep. We spend 1/3 of our lives sleeping, yet we do not know why or how.